Okay, good morning, everyone. We are very pleased to have with us on the forum of summer internship today, Dr. Ranjan Bhargav. I'll give you a brief introduction of Sir. Sir has done PhD from Department of Biotechnology, National Institute of Technology, Durgapur, West Bengal in year 2012. After that, Sir has worked as young scientist from Molecular Biophysics Unit, Indian Institute of Science, Bangalore, from a duration of 2 years. Then he has been associated as a research scholar from Department of Chemistry, University of Cambridge, United Kingdom, from 2015 to 2016. Sir has been working as a pool research scientist from Department of Biophysics, All India Institute of Medical Sciences from year 2017 to till date. He is also associated as visiting faculty of Central University, Jamia Millia Islamia, New Delhi. His area of research includes computational drug discovery, molecular dynamics, simulations of biomolecules, structural bioinformatics, and computational biophysics. He has authored many national and international research papers in SCI and Scopus journals, and has also been associated in many books of Elsevier. He is currently serving as reviewer of several international journals and as a member of editorial board. We are very pleased to have you on this forum, sir, and he will give insights on molecular dynamics simulations and their applications in drug discovery. Welcome you, sir.
Okay, so I will start.
Yes, sir.
Okay, thank you very much, Bairagya. Today, I will discuss about the molecular dynamics simulation and its application in drug discovery. Presently, I am working at AIIMS Department of Biophysics as a scientist in school. My area of research is computational biophysics, basically in molecular dynamics simulations. I quoted one line, in the real world, this would be eventually mean the most chemical experiments are conducted inside the in silicon of chip instead of glassware. So turn off the Bunsen burner, it will not be wanted for 10 years. This is not my statement, this is the statement from 1998 Nobel Prize winner chemistries. So initially, the MD simulation study is not appreciated by experimental community because they are thinking MD simulation or computational work, they have produced some large data that is not implemented in experimental study. So that time, before 2013, it's very difficult to publish the work on MD simulation and computational chemistry works. But the three Nobel laureate, Professor Martin Karplus, Michael Levitt, and Arces Wurzel, they got the Nobel in 2013 in the area of molecular dynamics simulations. Their area of research is the development of multi-scale model of complex chemical system in the area of computational biophysics. Professor Martin Karplus from USA and Professor Michael Levitt from our department, chemistry in University of Cambridge. They are all theoretical chemists. They are basically developed the molecular dynamic simulation methods, its applications, and they show that the experimental community and the theoretical community, both the data are similar and they are correlated to each other. Here I am showing what is molecular dynamic simulation. There is some video just to show that how, if you see here, how the protein is simulated, its change is conformation. If you see here, and as well as the residues, the entire protein is solvated. That's mean water molecule is there, and protein, the side chain is moving from one place to other place. If you see the upper panel, if you see the how the tyrosine and other residues change its conformations, this is very interesting. MD simulation study can only provide you the view of the structures. X-ray structure, NMR structure, they will give you only one snapshot. But a MD study give you a multiple snapshot and you can trace its conformations from native to other places. And this, if you see the left panel, this left panel water dynamics, that is the real, real human systems. Because this protein with water molecules and others, they are real event in our human body cells. That's why they got the Nobel. So how the protein is dynamics or dynamics is happen, their study is most interesting and their science is also is more interesting. So what is the historical background? Basically, the Rahman first done the MD simulation on liquid argon. After that, the Professor Macomb, he is the first professor who do MD simulation on protein. So this is basically his background. So at first we should know what is MD simulation. MD simulation is one type of method by which you can simulate or you can apply the force on any molecule. Due to that forces, the molecule will move its place from one position to other positions and within the dependent time scale. That's why it produced one good three-dimensional structures. They are basically provided two scope, one for microscopic, another for microscopic, and another for macroscopic. The macroscopic is basically for hydrogen bonding interaction or solvation free energy like that. So how the MD simulation is going, if you see how many phenomena is there, like polypeptide folding, biomolecular association, suppose protein-protein association, protein-DNA association like that. So for this reason, 1 to 7, you can do some MD simulation study. So MD simulation generally 2 to 3 types. One is classical MD simulation, another for quantum MD simulation. Quantum MD simulation, you can calculate the electron transfer mechanism from how the electron will transfer from one place to other place, or enzyme substrate complex, how the enzyme and substrate co-complex to each other and produce the product and their pathway from enzyme substrate to enzyme product. So this is chemical transactions or their chemical path you can trace by using quantum dynamics. This is actually short theory for MD simulations. Suppose if you have one atom, if you push the atom from one place to other place, you have implemented one force, that is F. F is equal to ma, that is force in mass into acceleration. It follows the Newton's rules of law. So minus dE by dr. So that is your rules. So if your atom is here, the position is, if you think T, E is go to forward position from other places. So that will be T plus delta T. And if it's the reverse position, T minus delta T in the respect of time. That's why we produce calculation the forces and updated its coordinate. If you take this R is the distance, T plus delta T, that's mean position from forward one place to other place, T minus delta T, that's mean its backward position. And this, if you do the summation, so this type of algorithm is followed by MD simulation method, that is called Verlet algorithm. Different type of algorithm is there, Verlet algorithm, Lipfroth algorithm, Verlet velocity algorithm, like that. It will follow the MD simulation. Those students who have a strong background in physics and chemistry, they will definitely do some MD simulation study and its applications. So this is the covalent bond angle. So when we apply the forces of any atom, it can be moved from one place to other place, or its bond will be stretching from like that one to other places. So in that places, CH3, that is the bond length. They can move from one place to other place like that. So in that cases, E bond is equal to KB into R minus R0. So that is the equation it's follow. As well as their angle, if you see their angle, how their angle is moving, they have some angle determination process. E angle is equal to K theta 0, theta minus theta 0. Also dihedral angle. Dihedral angle basically followed for 4 atoms. Generally for protein side chain, yeah, backbone atom, they will follow the dihedral angle. And other for non-bonded interaction. Non-bonded interaction are different types like improper angle, Van der Waals interaction, and electrostatic interactions. Generally, what we are doing, this bond angle and this dihedral angle, they are called bonding interactions, and non-bonded interaction is Van der Waals interaction and electrostatic interaction, as you know. And in cases the electrostatic charge of the water molecule will taken as 80, and for protein we take as a 4. So what is the force field? So that is called force field. One place to another place, and it will follow one potential energy, that is called bond angle, dihedral angle, improper, like that. So it is called potential energy. This potential energy will calculated by different force field, sometimes amber, sometimes charm, or different type of force field is there. So that is the method. So you can use amber or charm, whatever, for protein dynamics or protein minimizations. Next is solvation. Why solvation is required? Suppose if you take one protein, it should be surrounded by water molecules and other parts. So your system should be solvated in our human body cells. But in some computational chemists, they are also doing some calculation in without solvent. So solvation are two types. Generally, we are doing one for explicit solvent, another for implicit solvent. Explicit solvent, if you see the right side figure here, the water molecule you can visualize, and this water molecule is interact with the glutamic acid like that, so that you can visualize the water molecules here. That is called explicit. So there is two process for explicit solvation, one for stochastic boundary condition, another for PBC. And next is implicit solvation. Implicit solvation required for binding free energy calculation of protein or protein ligand like that. In that cases, the water molecule will be there, but you can visualize that water molecule, their charge and occupancy will be there for binding free energy calculations. For details, you see these explicit solvations. There is two types, one for stochastic boundary condition, another for periodic boundary condition. In stochastic boundary condition, if you see the left panel figures, the part of the protein, basically the beta helix, this helix is surrounded by water molecules, and that water molecules have been applied some forces so that this spherical shape of the water molecule cluster will be compact. So that is one method, that is called stochastic boundary condition. But we never use this stochastic boundary condition, we use the PBC. In PBC, the entire protein is solvated by 10,000 or 20,000 atoms so that it can mimic our body cell protein, this system. And in this PBC, the surrounding system, if you see, there is some rectangular box or cubical box like that. But after simulation, this box will be distorted because we never applied the force of the outside of the water molecules. This is the process of implicit solvation method. This is just for theory. The two methods had been followed, Poisson-Boltzmann and GB model. This method is used for calculation of binding free energy of ligand from MD simulation studies. There is two files is required for MD simulation, one for topology file and another for parameter files. What is the topology file? Topology file, when we do some MD simulation, the protein basically its residue change its file format. Suppose here you take the alanine, the entire charge of the alanine is 0. That is called total charge. This is the atom name. And if you see here, that is the atom charge of that particular position. And IC means internal coordinate. And here is this improper angle. So they change this format from one position to other position. And this is the parameter files. These parameter files require for MD simulation. If you see here, this position, this KB and R0, they bond angle, bond distance, dihedral angle, they will calculate it as per these rules. This MD simulation I have done during my research work. The protein is human INPDH. I take this protein and I do some MD simulation calculation. That's why I solvated this protein by cubical box, and we added sodium chloride or calcium or KCl so that the entire system of the protein should be neutralized. So this protocol of MD simulation will be followed, but it will depend from lab to lab and as well as system to system. Generally, what I am doing, energy minimization, heating and equilibration. These three steps is required, and finally for production run. Energy minimization, if you see here, basically the solvated molecules, basically water molecules and side chain of the water molecules like that. Heating is required from 273 to 300 Kelvin for two systems. Equilibration is three types. I am doing NPT, NVT and NPT. That is called ensemble. What is ensemble and how it is required this type of ensemble? If you see my slide, this is the flow chart of MD simulations. If you see the thermodynamic ensemble, that NVT, N is equal to, as you know, number of particle, V for volume, E for energy like that. If you come to this position here, there is three type of macrocanonical ensemble, canonical ensemble and constant NPT. So why it is required? For NVE, for microcanonical ensemble, here is this algorithm is required. Varlet-Leapfrog algorithm, velocity Varlet algorithm. And for canonical ensemble, your temperature will be fixed. To fix the temperature, you can use Berenson thermometer or noise overmeter. And there is another method is NPT, that is called isobaric ensemble. So this type of three ensemble is generally used. So what data you can take for dynamical behavior of the proteins? There is three data. One for local motion, another for rigid body motion, another for large scale motion. In local motion, There is three type of data you can obtain. One for atomic fluctuation, side chain motion, and loop motion. If you see the right side here, how the residues, the side chain is moving from one place to other places, that is called local motion. But in that cases, the displacement of residue is from 0.01 to 5 angstrom. Rigid body motion, that is helix or domain or subunit. Here is, if you can check the helix can move from one place to other place. So in case required, it around 10 angstrom displacement of the rigid body. So in that case, you have to run at least 1 second dynamics, but 1 second dynamics is very high. That means it's required almost 5 to 6 months. Another for large scale molecular dynamic simulation, suppose a structural transition from helix to coil or coil to helix, like association, dissociation, like protein like an association, dissociation like that, or protein folding, unfolding process. This scale you require for large scale molecular motions. And here the displacement of the residues is around 5 angstrom. So this type of dynamical phenomena you can obtain. For this large type of molecular dynamic simulation study, we used GPU, that is graphical process unit, that is high-speed GPU based. This GPU is highly priceable and it is also appreciated for MD simulations. Suppose if you have 10,000 atom, if you wish to run by CPU, 5 CPU, it takes around 1 to 2 days. But if you use the GPU, it takes less time. Within 5 to 6 hours, you can get the results. That's why GPU is generally used. This is the process of parallelizations of simulation in different processes. This type of high performance computing is required. If you see the left panel, this is called supercomputer or cluster. There is some 100 node is there. From 100 nodes, one node is equivalent to 5 or 10 CPU. So around, if you have some molecules to wish to run the supercomputer facility, so around 250 CPU is required to run good simulations. So after simulation running from the supercomputer, you can fetch this data from your supercomputer to your host computers. So your host computer should have good configuration, maybe GPU based or high core, like that means 32 core or like that. So this type of simulation facility is required for running or analysis the MD simulations. So there is some different type of method is there for softwares like Charm, Ember, Gromos, NMD like that. I am working on NMD with Charm force field, but you can also use for Gromos or Charm. But Ember is highly priceable, but in academic sector it is be less price. So other simulation package is also available. You can use it. Basically, the biotechnology student and computational chemistry student as required for this type of MD simulation study. Next is quantum MD simulations. That is slightly tough for student to understand this QM calculation. Generally, QM calculation is done by 2 or 3 methods like ab initio molecular orbital theory, semi-empirical method, and density functional theory. This density functional theory, you can use the wave function. This density functional theory you can use for stability, to check the stability of your system. But in that cases, I told you the large biomolecular system is not appreciated. The small molecule like your ligand, your 3 or 4 residue will be there. You can do DFT calculation or electron like that. You can check its stability or binding free energy, and you can compare this binding free energy with your MD data. That is the birth of the quantum mechanics. Professor Einstein, as you know, is equal to h nu, and nu is equal to c by lambda. If you see here, the nucleus is there. From nucleus, if you electron jump from one place to other place, the energy in release del E is equal to h nu, and this concept is implemented for computational facility, and they use H is equal to E psi. So H is equal to Hamiltonian operator. You can calculate the kinetic energy of the nucleus and kinetic energy of the electron, and this is the method for calculations. These are few methods like molecular mechanics, semi-empirical, ab initio method. By using the small molecule or ligand with some residue, you can calculate their stability, binding energy, or you can check its chemical path, transition path from one place to other place. That is most interesting, but it is only applicable for small molecule. But large molecule cases, molecular mechanics and MD is most appreciated. Nowadays, the MD simulation is going from quantum mechanics to mesoscale level. So QM all atom is doing is previous, but nowadays the people are most interested about the coarse grain dynamics. This coarse grain dynamics very interesting. In that case, what you have done, protein folding, unfolding process, global motion, generally G protein coupled receptor. If you wish to do some MD simulation with coarse grain modeling, you can use the protein-protein interaction study and how the proteins interact to each other, or protein ligand associations. This type of coarse grain dynamics is used nowadays, and the mesoscale level dynamics also used. Suppose you have a drug, and how the drug will behave within the human body. If you do some mesoscale level dynamics, it will give you some good results, and this results is definitely appreciated by medical science. So that is your main aim, from quantum to all atom to coarse grain to mesoscale level. So that is very interesting. I encourage all of the students to do some MD calculations before doing some experiment study. What is its application? Its application is you can check the stability of your protein, conformational changes, protein folding, or molecular recognition like that. Here I am showing few of my work which is applied by MD simulation and MD simulation based drug discovery. So these three slides I will show you. Number one, if you see here the IMPDH, that is the one protein which contain the substrate binding site and another for cofactor binding site. Within this binding site, I have observed these three water molecules, W722, W646, and W611. These three water molecules is not available in crystal structure. During MD simulation, I have found these three water molecules is interesting and interact with each other, and from this point of view I suggested that three water molecule is appreciated for water-based drug discovery. Basically is WM. They have some 8-centered hydrogen bond, and they are most interesting, and they have some role for possible drug discovery. But how it will use for drug discovery, I will show you in later slide. Then loop flap dynamics. Suppose if you have these two loop region, one region and another region, these two regions have been stabilized by ligand. But if you remove this ligand, how these two loop region will stabilize to each other? So if we do some MD simulation, then I have found some water molecules, they have stabilized these two loop region by 4 water molecules. So from this point of view, I suggest these 4 water molecules have some stabilization role. And this is the cryptic binding site. This cryptic binding site is generally occurred during MD simulation time. It is not available in any X-ray or NMR structures, and this is not a substrate or cofactor binding pocket. This is the new ligand binding pocket. This volume of this pocket is around 540 angstrom cube. You may design some drug and put this drug here so that it may act as a good inhibitor. Next is water-based drug discovery. How the water-based drug discovery is happening. In the same case, I take the substrate binding site as the chloro-IMP CPR, which attach the covalent to CPR. You observe these two water molecules here, W7 and W9. So I am thinking, I wish to utilize the position of W19 and W7, and then I use CH2OH group is here and another for here, W7 and W9 position. In this position, so that it will replace the position of W19 and W7. So in this position, I have observed that CH2OH, I just added this yellow line here. You are seeing the CH2OH is added, and the oxygen atom and this oxygen atom is around 0.5 angstrom. And then I ran some MD simulation study, and I observed more number of water molecules is occupy the position. So that is called sidechain modification of the drug on the basis of MD simulation study. So this type of work we can approach on the basis of MD simulation, but in X-ray or NMR data, it is very impossible to do some new drug on for structure-based drug discovery. My next part is computer-aided drug design. Computer-aided drug design, as you know, most interesting cases. Nowadays, computational drug design is most appreciated by experimental scientists before going to do some experiment or synthesis of like that. So if you have some computational data, you suggest something and it will guide you a new thing. So computational drug design is a new method suppose you have the target region, and you do not know how the drug will bind to this position and which parameter is there, you do some CAD method. So what is the role of CAD method? Generally, few steps is followed for drug discovery, like safety, efficiency, stability, solubility, synthetic availability, and novelty. So this CAD is required this type of parameters. And here I am showing you how the protein structure is evaluated and how do you identify the lead compound for drug discovery. Suppose if you take this isolated protein, this protein is here, but you do not know the actual ligand or actual drug, how it will bind. So this is the structure. This structure, if this structure is available in protein database, by using X-ray or NMR study, you can obtain this data. Else you have do some molecular modeling technique or homology modeling technique and designs this drug on the respect of this geometry, this position. Else you can do some virtual screening. What is virtual screening? Virtual screening is one type of method. Suppose you have some database like n number of compound. These compound are subdivided in different places, like FDA approved drug, non-FDA approved drug, some other inhibitors, some Cambridge chemical database inhibitors, like that. They've categorized. You take this ligand and fit this position and check its binding affinity. That is called hits. So this hits is going called virtual screening. On this basis of hits, you can identify the best ligand, and then you can do some calculations for experimental study. So steps involved in drug design, as you know, target identification and validation. That's mean if you have the receptor, you have identified the receptor, and validate is receptor structures. Basically is geometry in active site pocket. Next is lead identification and optimization. Lead means there is two parts of the drug, one for main compound and another for subsidiary compound. So main compound is directly interact to the active site binding pocket and inhibit this position, and other part that helps this drug to move into the cell, that is called a Y part. So this lead compound is very interesting. We generally design this lead compound, and then we try to do some molecular docking and other simulation part, and then, as you know, preclinical trial or clinical trial, like that. This is the method for molecular docking. One molecule is there, you wish to try to dock here, you can do docking by manually, by hand, or by computational program. So you should know what is molecular docking. Molecular docking is a technique by which you can place the ligand in the active site pocket, and so that both the conformation, if you have the ligand here and substrate here, so this receptor and ligand bind to each other, and they are compared to each other like more stable condition with more stable conformations, and their binding energy will be more stable. So this type of conformation is checking for experimental study. So molecular docking generally used for unknown cases. Suppose if you have the protein ligand complexes in X-ray structure, but its comparative study, you need some new ligand, but you do not know how it will behave to the same receptor. That time you can also take these molecular docking techniques. Or I would suggest, other than molecular docking, you can do some MD calculation, then try to docking. That will be more appreciable. More computational demanding and accurate procedure can be applied for docking of a single ligand or receptor or more ligand. Docking is an established methodology in structure-based drug design, as well as increasing the importance of the explanation of results from structural genomics. What I am trying to say that you have the target structure here, and this structure will maybe obtained from X-ray or NMR or homology model. And here is the molecular library which is available for databases. You can take this ligand from here and try to dock. If it is fitted well, then you do some ADMET test and go for other experiment. So that is actually drug discovery procedure. For this type, as you know, this drug in the cathedral, that is the drug which we taken as a COVID drug. This is the M protein, most responsible protein for coronavirus, which is found inside the cytoplasm, and that is this cysteine 145, histidine is there, and we try to dock this indicatorol to this position so that we can try to check how it will behave within active site pocket. So this study is going with my student, and they have identified this is that may be act as a good inhibitors for these positions. So there is 3 parameters. One is identification of ligand binding site on the target macromolecule is this position. Sampling of possible geometric arrangement of the ligand and receptor. Geometry means it will properly fit to each other, and scoring function. So what is scoring function? Scoring function is generally here is taken as non-bonded interactions like hydrogen bonding interaction, Van der Rohe interaction, electrostatic interaction, plus other part, solvation energy. This entire term is called scoring function. If this scoring function have more negative, that's mean more suitable, and they will strongly attach to the binding positions. The binding site of the protein of small molecule or DNA or RNA can often be identified by following 3 methods: this putative structure, putative docking, followed by structure-based homology modeling. But here I will show you some drug related to computational drug discovery, and which is finally discovered by experimental scientists. So sampling of molecular docking, there is 2 types of molecular docking is used, one for rigid body docking and another for flexible body docking. Rigid body docking, in case, suppose you have the receptor and this is the ligand, and this receptor, the ligand is directly come and bind to this position without any effective side chain or rotatable bonds will be fixed. So there is nothing any change. That is called rigid body docking. This rigid body docking is initially doing for structure prediction. Generally, crystallographer use this rigid body docking. Whereas flexible body docking, after rigid body docking, you can do some flexible. That's when your flexible bond, your rotatable bond will be rotate to each other and flex and properly fit to this position. That is called flexible body docking. And in this case, the rigid body docking, there is 2 method is used, translation and rotation. But in flexible body, there is 6-dimensional uses, both ligand and receptor. For clinical application of your molecular docking study, I use some some of the example where the drug is evolved from CAD. Like one, HIV protease. As you know, HIV protease, the drug is evolved by using CAD. This is the HIV protease. This is the structure of HIV protease. N-terminal, this is the C-terminal, aspartic acid, and these are the active sites. So what they are doing, if you go to the previous slide, there is a 20,000 compounded binding database. They took screening 200, and from there they selected base 16. So what they are doing, they take this drug, indanavid drug, this part they have identified, this part without this amine group. This part they have identified and fit this position. After that, they are thinking if they added this amine group with benzene ring, so that will be more suitable. So after that, they can add and do this molecular docking techniques and evolve this indanavid drug. So this drug is basically coming from CAD. This is one example. Another example is human carbonic anhydrase. This is a good anti-glaucoma and anti-tumor drug. If you take here, this is the carbonic anhydrase. These are the drugs they are evolved during CAD. So they use the FLEX program. From there, the 90,000 compound databases there. From there, 13 hits they are calculated. 4 molecules they have chosen or selected as a best drug candidate, and these 4 molecules is better to other existing drug, like this amide 2 drug is here. They have compared this 4 compound and with this 2 amide drug, and they have calculated their KD value in nanomolar scale, and they observe this is that will be good binding positions, and these 4 compound they pick up the one drug that is most interesting. So this is one case, and another case which evolved during CAD study. Next I am showing me and my student is working is COVID protein, like cancer, COVID protein, this coronavirus, as you know, this is the structure of spine, and this is the M protein is here in the nucleus. So these two protein is most interesting is main protease, and another for is RBD. RBD is main receptor binding domain. They are observed in spike position here, and this is also here. So what I am doing, how the HCQ, that's mean hydroxychloroquine, interact to each other. So it will bind to RBD or main protease. For this reason, we take these two and do some molecular docking study, and I have found this M protease is more suitable to attach the HCQ, that's in hydroxychloroquine. This hydroxychloroquine HCQ, they attach to this cysteine 145. If you see here this thiol group, they attach to this chlorine. This is most interesting, and also histidine 41. So they will attach to these impro binding positions with good binding affinity. So this work is going on in with my student. So the application of MD simulation for drug discovery, that is the entire thing. That is the entire thing. Number one, structure, frame, and simulation. Suppose if you have some structure, you do some MD simulation study without ligand, so that it have some different conformation, maybe open conformation or closed conformation. So this is a step here. Next, you can doing here as a clustering process. This clustering process, you can obtain the structure in different time scale. If you see the right panel here, so different structure is there. On the basis of different structure and their conformation, you do some virtual screening and design some ligand and predict the ligand, which is most interesting. So this type of work actually implemented in MD simulation based drug discovery. And that is most appreciated in most of the biotechnology and computational chemistry and biophysics researchers are doing this type of drug discovery. So the therapeutic concept that if you have some drug, so how the drug will interact with each others in human body cells. If you do some mesoscale level dynamics, that will be most appreciated. But you should remember its side effect and toxicity, and also ADMET test. How, if the drug have side effect or not, or toxicity is high, appreciable or not. So this type of information is required before going to propose this is a good drug or not. Preclinical trial, as you know, this is the three method is there: absorption, distribution, metabolism, excretion, and toxicology, and followed by Lipinski rules of 5. If it is follows, you can predict that it is a good drug. So that is the method for virtual screening based virtual screening based drug discovery, and this chemoinformatics process, how the drug is designed on the basis of this process. And as you know, a few clinical trial is required, like phase 1, phase 2, phase 3, or phase 4 before going to predict as a good drug or not. But what is actual role of biotechnology industry? As you know, there is a good step is there, followed by a different step like drug discovery, product development, process development, bioanalytical development. Like these are the followed, and they have categorized into 3 parts. One is drug development and discovery, next for operation, and another for commercialization. So what is the actual role for simulation scientist or MD simulation specialist? You have some role in drug discovery and development part. This part you can design, you can design this drug discovery part, and then process your drug, followed by operation methods. But in operational methods, some data scientists are required for analysis their data and other process. And commercialization, it is basically for sales people. So the present status of simulation of biotech industry is 4. Number 1, to identify drug candidate. Number 2, to analyze the human genome for disease trigger, to stimulate the behavior of drug molecule in human body, to stimulate the manufacturing process and mass balance. So the present status of the simulation. And I am very much thankful to my few students. They are working with me last couple of years, and they also published good papers. Thank you. Any question?
Yes, students, any question? Sir, you can stop sharing the screen.
Stop sharing.
Yes.